Hello students, welcome back. In this particular series of principles of agronomy, uh, we have so far covered about soil and its effect on soil management or soil environment and its effect on crop growth and development. So in particular soil environment and its effect on crop growth and development, we were discussed about so far like soil physical environment and how it is affecting the crop growth. Soil chemical environment, in previous lecture we were discussing about soil chemical environment and what are the parameters coming under soil chemical environment and how it is affecting the crop growth and development. Now today we are going to discuss about soil biological environment, how it is affecting the soil properties as well as crop growth. Okay, so as in the previous lecture also I was talking about most of the part most of the important part of the plant, and that is a root system. So major part through which it provides support as well as takes of water and nutrient for the growth and development. The root portion or rhizospheric portion, it belongs to soil room. Okay, soil zone. So soil biological property, how it is affecting the crop growth, right? See, soil biological properties means soil microbes. Basically, it indicates to soil microbes. Okay. So when we're talking about soil microbes, they have very significant role in soil. Okay. So some microbes they do nitrogen fixation. Some microbes they are helpful in toxic material decomposition of toxic material or simplification of non-toxic form. It converts the toxic form to non-toxic form. Okay. Some soil microbes they helps in nutrient transformation. That means unavailable nutrient to available form. Okay, like that there are various role of soil microbes. If we we talk about, if we classify soil organisms, okay, so basically soil biological environment that depends on that is a major function of soil organism. So soil organism can be grouped into soil flora and soil fauna. Under soil flora there are macrofloras are present and microflora are present. Under soil fauna as well, macrofauna and microfauna are present. So what are the examples of macroflora? Macroflora are the root of higher plants. Okay. If we talk about plants and their roots, they are coming under macroflora. And soil microflora, it is like bacteria, fungus, and actinomycetes. They are coming under soil microflora. Then macrofauna, these are earthworms, moles, ants. These are called macrofauna. They are present in soil. And microfauna like protozoa, nematode, the micro, animals or micro, you know, things, they are called microfauna. So microflora are bacteria, fungus, and actinomycetes. Microfauna, protozoa, nematode. So you have to remember this table. Basically these four examples like macroflora, roots of higher plants is coming under macroflora. Then macrofauna, earthworm, moles, and ants, they are coming under macrofauna. Microflora, bacteria, fungus, and actinomycetes, they are coming under microfauna flora. And microfauna, protozoa, and nematode, they are coming under soil microflora, right? Now how these soil microorganisms, I mean, if we're talking about rhizosphere means the microenvironment that is covering the roots of plants. Okay, that is rhizosphere zone. Okay, exudation of energy rich compound. So basically how these microbes they live in the rhizosphere. Basically plant root it secretes some high energy compound or carbonaceous compound which helps the microbes to grow. That means microbes, as like human being or animals, microbes also as well as plants, microbes also need some source of energy, I mean, so some source of food. So what is the food of microbes? For microbes the food is the soil organic matter or soil organic carbon. Carbon is the food for microbes. And when some exudates are being secreted to the soil through rhizosphere, or I mean root system, so that helps the microbes to grow there, right? And let me tell you, 7 billion bacteria you can find in 1 gram soil of rhizosphere. Okay, and if we'll talk about other soils, there are around 25 to 30 millions of bacteria you can find. So rhizosphere zone is the epicenter of the microbial activity. That means rhizosphere secretes some chemical or some compounds which are carbonaceous and which are energetic compounds. So microbes feed to those compounds, and they can get feed in the near the rhizospheric zone. So their dominance is higher near the rhizospheric zone, right? Based on oxygen, microbes are categorized into obligate aerobe, facultative aerobe, and anaerobe. So what is the difference between this obligate aerobe and facultative aerobe? So in case of obligate aerobe, these microbes, they require oxygen, and without oxygen they cannot survive. Okay. Then facultative aerobe, they require oxygen, but without the oxygen they can survive. Okay. Anaerobe, they doesn't require any oxygen, okay, for their survival. They can even survive in anaerobic condition. Then based on nutrition, there are autotrophs. Based on nutrition, autotrophs and heterotrophs. Autotrophs means they can, you know, synthesize, or they can, have their own food. Hetero Heterotrophs means they depends on other organism, other living organism for their nutrition. Okay. Autotrophs can be also two types. One is photoautotrophs, the microbes who can use, sunlight or solar radiation to synthesize their own food. They are called photoautotrophs. Chemoautotrophs means they can use energy from the chemicals or from the minerals that is present in the soil. That is chemoautotrophs. So this is how microbes are being classified. One is broad classification like flora, fauna. Another one is based on oxygen requirement, based on nutrition. These are being classified. Again, based on temperature. Based on temperature requirement, microbes are classified into three different broad groups, like psychrophile, uh, which microbes they require less than 10 degrees Celsius, and mostly they are found in, temperate zone. Then mesophiles, the requirement of temperature is around 20 to 40 degrees Celsius. Thermophile, they can survive beyond 40 degrees Celsius. And if we'll talk about, different microflora in the soil like bacteria, fungus, and actinomycetes is mainly dominated in dry condition or dry soil. Optimum moisture for aerobic bacteria, that is 50 to 70% of water holding capacity. Most microflora are mesophilic. Okay, mesophilic means the temperature ranges from 20 to 40 degrees Celsius or 15 to 45 degrees Celsius. And optimum temperature is around 37 degrees Celsius. Higher carbon dioxide concentration is toxic to microbes. Even direct sunlight is injurious to microflora. Okay, pH. If we look at pH, the bacteria for bacteria survival, pH, neutral pH is mostly, optimum 6.5 to 8. That is optimum. For fungus, acidic pH is optimum like 4.5 to 6.5, and for actinomycetes, slightly alkaline soil is, optimum. Okay. Then let's discuss about some soil bacteria. Soil bacteria examples like Pseudomonas, Azotobacter, Bacillus, Arthrobacter, Aerobacter, Agrobacterium, Rhizobium, Clostridium, Beijeringia, Nitrosomonas, Nitrobacter. These are some important soil bacteria which help in different, nutrient transformation in the soil. Then highest bacteria is having the highest population among the microflora. If we look at microflora like fungus, bacteria, and actinomycetes in soil, then in 1 gram soil, if we take, most dominant microflora is bacteria in terms of number. Okay. Now coming to soil fungi or fungus, they prefer acidic soil pH, and we have already discussed about it. Mostly 4.5 to 6.5 pH they require. Then they are larger in size than bacteria, and they are having the highest biomass among the microflora in the soil. Okay. If we'll to take 1 gram biomass, and if we'll ask what is the weight of microflora which is having the dominance, then you can blindly answer fungus. They can also decompose lignin. Lignin cannot be decomposed by bacteria, but fungus can decompose lignin. For some examples of common fungus found in soil, like Aspergillus, Fusarium, Trichoderma, Mucor, Penicillium, Mycorrhiza. That is the, fungal association with root of higher plants, like Ectomycorrhiza and Endomycorrhiza. Ectomycorrhiza examples are Boletus and Amanita. Endomycorrhiza examples are Phoma and Rhizoctonia. You have to remember this. Then actinomycetes. This is another group of, microflora. Their size varies between bacteria and fungus, and population also second to bacteria. They, they usually the decomposition rate by actinomycetes is usually slower than bacteria and fungus. Example, streptomyces. Then another group of microorganism found in soil, that is algae. Algae are mainly autotrophic, and there are different types of algae are found. One is blue green algae, that is called cyanophyta, and it is mainly found in tropical region, and it is most common genera are nostoc and anamina. Okay, that is blue green algae. Then grass green algae, that is called chlorophyta. Mainly you can find them in grasslands of temperate region. Then yellow green algae or xanthophyta. Then bacillariophyta or golden green algae. These are less abundant. Yellow green algae and golden green algae, they are less abundant. Okay, most abundant is blue green algae, which is mainly found in tropical region. And in temperate region, you can find grass green algae. Microbes or microorganisms, they secrete several enzymes like urease, phosphatase, cellulase, glucosidase, dehydrogenase. These actually help in break down some enzymes. They help in breakdown of organic matter. Some enzymes, they help in nutrient transformation. Some enzymes, they help in actually, in organic matter there are different biomolecules found, right? So for different biomolecules decomposition or disintegration, different types of enzymes are being secreted by different microbes. Okay. Rhizobium. Rhizobium, it secretes enzyme nitrogenase. Okay, it is a heterotrophic aerobic bacteria. Okay, rhizobium, it helps in nitrogen fixation, right? Effective nodulation, if we will talk about effective nodulation, actually rhizobium is it forms nodule in the root of leguminous plant. And when we call a nodule effective, it must have higher leg hemoglobin content, and it is having larger diameter, and it is pinkish to reddish in color. Right, leg hemoglobin is having higher affinity to oxygen, so it is helpful in oxygen transformation. High nitrogen, low phosphorus. If the nitrogen content of soil is high and low phosphorus in soil, then biological nitrogen fixation is less. Rhizobium, it is symbiotic nitrogen fixing bacteria. Then Azotobacter, Azospirillum, Clostridium, Rhodospirillum, these are non-symbiotic in nature. That means they do not, help each other. That means they are free living. Okay, they do not help. I mean, need a host plant for their action. That is non-symbiotic in nature. Soil organic matter, if we'll talk about soil organic matter, it is found less in cultivated soil or conventionally tilled soil, because if when we cultivate the soil, there is liberation of carbon to atmosphere as carbon dioxide, so carbon oxidation happens. So soil organic matter is quickly decomposed, and it is liberated as carbon dioxide into the atmosphere, if we are going for conventional tillage or cultivation. Soil organic matter is the sink, it is kind of sink to atmospheric carbon dioxide. Okay, so when and there are different polypeptides found in soil organic matter, which are uh, in the process of aminization they convert into amino acid. Then the process of ammonification, they convert to ammonium. Then through nitrification process, they convert to nitrite and nitrate. Like here, the use of bacteria are from ammonium to nitrite. For conversion of ammonium to nitrite, the bacteria Nitrosomonas helps, and from nitrite to nitrate, the bacteria Nitrobacter helps. And for nitrification process, the optimum pH is 6.5 to 7.5, and optimum temperature you can find 30 to 35 degrees Celsius. Then there is a process of denitrification happen, where nitrate is converted to nitrogen gas through Bacillus pseudomonas. Okay, that is called denitrification, and we have already talked about it. Then if we'll talk about soil organic matter, what are the constituent of soil organic matter? So soil organic matter, it simplifies through hydrolysis. And what are the constituent of soil organic matter? If we'll talk, that is cellulose. Soil organic matter mainly composed of cellulose. 20 to 30% cellulose you can find. Hemicellulose you can find 10 to 30%. Lignin also you can find 10 to 30%. Protein around 1 to 15%. Fat and wax around another 1 to 8%. Cellulose decomposing bacteria, some cellulose decomposing bacteria you can find in soil like cytophaga, sporocytophaga, uh, argioko. Cause Polyangium, Clostridium. These are some cellulose decomposing bacteria. So there are two concepts of nitrogen dynamics. One is mineralization of nitrogen. In case of nitrogen as well as phosphorus, you can find this concept like mineralization and immobilization. So when the CN ratio of whatever organic matter we are adding, if it is less than 20 is to 1, then there will be mineralization. That means from complex organic matter to inorganic matter. Organic matter to inorganic, that means from unavailable form to available form. You can find if CN ratio is less than 20 is to 1, then there will be mineralization. When immobilization happen, when CN ratio is greater than 30 is to 1. Similarly, for phosphorus, when mineralization happen, when the CP ratio, it is less than 200 is to 1. When immobilization of phosphorus happen, when the CP ratio is more than or higher than 300 is to 1. Okay. Then critical for sulfur, the CS ratio is 50 is to 1. If it is less than 50 is to 1, then there will be mineralization. If it is more than 50 is to 1, there will be immobilization. For sulfur, there are two other dynamic mechanisms like sulfur reduction and oxidation. For sulfur reduction, desulfovibrio is the common microbe that plays a significant role. For sulfur oxidation, thiobacillus plays a significant role. Then modification of soil biological environment. Modification of soil biological environment. There is a term called metabiosis. Metabiosis means it is a form of ecological dependence in which one organism or a functional group of organisms must modify the environment before another organism or functional group of organism can live or thrive in it. Okay. So basically, one organism or a group of organisms, they modify the soil environment according to other organism or other group of organism before the other group of organism actually live in the soil. So this process is known as metabiosis, and metabiosis is done or modified by metabiont. So previously existing organisms, they are called metabiont. Okay. So types of metabolic actions like facilitation, ecological engineering, commensalism, keystone predation, these are some of the important metabiotic action which we will discuss in some other lectures. Okay. Now, if we're talking about agricultural soil and soil environment, there is one important aspect we have to look into, that is environmental changes in submerged soil. Okay, so when the soil is submerged, when our agriculture crop field or any soil is submerged, there are some changes in physical, chemical as well as biological properties of soil happens, which also affect crop growth and development. So mainly we can grow rice, paragrass in submerged soil because they are having developed parenchyma tissue has been developed in this type of crop, and they can aerate themselves through parenchyma tissue. Okay, which can take oxygen from the environment and it can feed that oxygen in the root system where respiration, so that root respiration can continue. What are the physical changes happen when there is submerged soil? The osmotic potential is around zero. Osmotic potential of water or total water potential is around zero. It increases BOD or biological oxygen demand. It reduces oxygen concentration. It increases the carbon dioxide level, and it forms bicarbonate, which is toxic. It reduces the soil strength. And when we are going for puddling, that means disturbance of soil in suboptimal or submerged condition, then it destroys soil structure as well. What are the chemical changes happen when there is submergence? pH, it tends to neutrality. Suppose the pH is earlier acidic, it will come to neutral. Suppose the pH is earlier saline or alkaline, it will also come to neutral. Increase in phosphorus, iron and manganese availability. More methane emission will be there. Denitrification process will increase. Reduction of, availability of copper and zinc. More H2S availability, which will become toxic, or hydrogen sulfide that becomes toxic. Then what are the biological property or biological changes if the soil is submerged? Non-symbiotic bacteria population will increase, denitrifying bacteria population will increase, then actinomycetes and fungal population will decrease. Actinomycetes and fungal population will decrease. So these are some of the changes in soil environment if the soil is submerged. Okay. So in this particular lecture we discussed about soil biological environment and how the soil property changes according to soil, somewhat condition, right? So if you have any doubt regarding this particular topic, any concept you have, if you have any doubt regarding particular to this what we have discussed today, or anything else from any part of agronomy or soil science, you can let me know in the comment section. So till then, keep reading and keep signing.